So, good morning to all of you students of BT first sem. Today, I am going to discuss about the cytoskeleton that is given in your cell biology syllabus. That is, we will talk about microfilaments, microtubules, and intermediate filaments. So, today we will mainly focus on microtubules, and in the next classes I will discuss about the microfilaments and intermediate filaments as well. So let's get started now. So about the cytoskeleton, if I say that, what can I say about cytoskeleton? The ability of eukaryotic cells to adopt a variety of shapes and to carry out coordinated and directed movement depends on cytoskeleton. So eukaryotic cells have different shapes. Like you can see the red blood cells have a unique shape. The nerve cells have a unique shape. The dendritic cells also have a unique shape. Okay, that is actually mediated by your the pillars of the cell, which is called as cytoskeleton. Even the movement also depends on cytoskeleton. Now, the cytoskeleton extend throughout the cytoplasm. It is spreaded throughout the cytoplasm, and it is a complex network of three types of protein filaments. Now, basically, three different types of protein filaments are involved in making this pillar, in making this, what can I say, the skeleton part of the cell. Now, what are those three protein filaments? Microtubules, microfilaments, also called as actin filaments. In many books, you will find they mentioned as actin filaments, and intermediate filaments. Okay, so together, cytoskeleton is composed of these three different types of protein filaments, made up of different proteins. Some are dimeric in nature, right? Another name of cytoskeleton is also called cytomusculature, because it helps in the movement like crawling of cells on a suitable substrate. It also helps in contraction and relaxation of muscles and many changes in the shape of a developing vertebrate embryo. Okay, so cytoskeleton, also known as cytomusculature, because it helps in the movement, different types of movement like crawling of cells on a suitable substrate, then muscle contraction, then your the different changes that we observe during the developing of a embryo in vertebrates. They are all done by the help of cytoskeleton protein filaments. Now, this cytoskeleton also provides the machinery for cyclosis in the cytoplasm. So, cytoskeleton also helps, also provide in the machinery of cyclosis in the cytoplasm. What is cyclosis? Cyclosis is the flow of cytoplasm inside the cell driven up by the forces from the cytoskeleton in order to speed up the transport of the molecules, and organelles around the cell. So cyclosis simply means the flow of the cytoplasm inside the cell. So cytoplasm is moving inside the cell, and it is driven by the forces from the help of cytoskeletal proteins. So cytoskeletal protein filaments helps in this movement, And why this type of movement occur inside the cell, inside the cytoplasm, with the help of cytoskeletal proteins? Because to speed up the transport of the molecules, to speed up the transport of the organelles around the cell. So when we say that the DNA or the RNA gets moved towards the cytoplasm, where the ribosomes grab on it and carries out the translation process, then we have seen the endocytosis, the exocytosis, all are actually driven by these cytoskeletal proteins. Okay, and the process is called cyclosis. Okay, so coming to this next point, I will not talk all the things here. The important one I will discuss here. Now, the main proteins that are present in the cytoskeleton. Now, we have said about the protein filaments before, that microtubules, microfilaments, and intermediate filaments are the main protein filaments found in compose the cytoskeleton. But what are the proteins that make up the microtubules? What are the proteins that make up the microfilaments, also called as actin filaments? And what are the proteins that are involved in making intermediate filaments? To make microtubules, tubulin proteins are involved. To make microtubules, tubulin proteins are involved. To make microfilament, we need proteins like actin, myosin, tropomyosin, and other proteins to make microfilaments. Okay, keratin, vimentin, desmin, and lamin. Okay, and others in case of intermediate filaments. So it is saying that to make intermediate filaments, we need keratin, we need vimentin, desmin, lamin, and other proteins are also there to make intermediate filaments. Now, uh, if we differentiate, if we try to see the structure of tubulin and actin, what it is saying here. So I think you have understood this part, what I have said, that to make the microtubules we need tubulin. To make microfilaments we need actin, myosin, tropomyosin, and others. For making intermediate filaments we need uh, keratins, vimentins, desmins, and lamins. Okay, now tubulin and actin are globular proteins. Tubulin and actin are globular proteins, while the subunits of intermediate filaments are fibrous proteins. What are globular proteins? I have mentioned in this book, but I will try to say here. Globular proteins are spherical type of proteins and are somewhat soluble in water, unlike the fibrous proteins or membrane proteins. So, tubulin and actins are spherical type of protein, globular in nature, and they are somewhat soluble in water, unlike fibrous proteins. While the subunits of intermediate filaments are fibrous proteins. So, this means lamin, keratin, they are fibrous protein. They are not soluble in water, right? And they are non-globular. Okay. So up to this much you should see and write as a notes. I will not write all the things here. Now we will discuss about the microtubules here now. Let us see about the microtubules. So I have said that microtubules is made up of tubulin. Okay. Microtubules were first observed in the exoplasm and myelinated nerve fibers by Robertis and Franchi. So Franchi and Robertis have first observed the structure of microtubules observed in the exoplasm within the axon, which are long thread-like part of nerve cell along which the nerve impulses are conducted from the cell's body to the other cells. So within that exoplasm, within the axon of the nerve fibers, Robertis and Franchi in 1953 have observed the microtubules. They first call them as neurotubules because they are found in the nerve cells. Same nature of microtubules were found by Sabatini, Bench, and Barnett in 1963. Okay, microtubules of the plant cells were also identified by Leadbetter and Porter in 1963. So microtubules is found almost every in plant cells and animal cells as well, right? Now, occurrence. So I have said that microtubules first observed by whom? Robertis and Franchi in 1953 in the myelinated nerve cells. Okay. Then it was found by the Barnett, Sabatini, and Bench on other type of cells as well, and in especially plant cells also microtubules were observed by Porter and Leadbetter in 1963. Now about the occurrence. With rare exceptions such as human erythrocytes, microtubules are found in all eukaryotic cells, either free in the cytoplasm or forming a part of centrioles, cilia, and flagella. So, what can I say? Here, it is saying that in some of the there are some exceptions also. There are some exceptions also that microtubules are not found, like in human erythrocytes. But in most of the cells in human, in eukaryotic cells, other type of eukaryotic cells have microtubules. Either they are found free in the cytoplasm or they form a part of centrioles, they form a part of cilia and flagella, which are needed for the movement of the cells. The most abundant source of microtubules for the biochemical studies is the vertebrate brain. If you want to really study about, if you want to see that where mostly this type of microtubules are found, it is found in the brain of vertebrates. High densities of microtubules exist in the axons and dendrites of the nerve cells. Microtubules are mostly found, abundantly found within the axons, within the dendrites of the nerve cell. I'm sure you know about the dendrites. You have learned dendrite are the short branch extension of a nerve cell along which impulses are received from the other nerve cells at the synaptic position and are transmitted to the cell body. So dendrites are found within the nerve cells, dendrites, remember, where the impulses are received. So they are also made up of microtubules. Even the axons of the nerve cells are also made up of microtubules. And most of the microtubules are found within that part. Okay. Now, in the cytoplasm of animal and plant cells, microtubules occur at 7 sites. So there are 7 important sites where microtubules are found abundantly. Okay, what are those sites? Cilia flagella, which helps in the movement of the cell. Well, long flagellar tail is found within the sperm cell. That's a big example. Centrioles and basal bodies. Well, centrioles and basal bodies also they are made up of the microtubules. What are basal bodies? Actually, basal bodies is a protein structure found at the base of eukaryotic uh, so basal bodies basal bodies are actually, it is a protein structure found at the base of eukaryotic unduly podium are the motile filamentous intracellular projection of eukaryotic cells. They are motile, means that they helps in movement. Filamentous intracellular projection, which are project inwards of a eukaryotic cell. Basal bodies generally are formed from a centriole and several additional protein structures, and essentially it is a modified centriole, you can say. So basal bodies contain this type of microtubule. Nerve processes, mitotic apparatus, obviously the spindle fiber is made up of microtubules. Cortex of meristematic plant cells, elongating cells such as during the formation of lens or during spermatogenesis of certain insects. Selected structures in protozoa such as axostele of parasitic flagellates, the axoneme of Echinosphaerium, right? The fiber system of stentor and cytopharyngeal basket of Nasula. So these are the some essential sites where most of the is filled up with microtubules actually. Okay. Now the stability of microtubule varies. Cytoplasmic and spindle microtubules are rather labile structures. That means they multiply continuously, whereas those microtubules which are found in cilia and flagella are more resistant to various treatment. The microtubules which make up the cilia and flagella, they are more stable, they are more resistant to various treatment, they will not get degraded so much quickly, while the cytoplasmic microtubule or spindle microtubule are rather labile structures. They multiply continuously. They can be treated with various chemical, like a chemical name called G-scene, which disrupts the microtubules assembly, right? Okay. So this was about the occurrence of the microtubule. Let us see now about the structure. So about the structure, it is saying that microtubule consists of a long, unbranched and hollow tubules 24 to 25 nanometer in diameter, several micrometers long, and with 6 nanometer thick wall having 13 subunits or protofilaments. So about the microtubule, if is say the structure of microtubule, it is a long like structure, unbranched. It does not have any branches. Inside it is hollow, and the diameter is about 24 to 25 nanometer in diameter. This microtubule is made up of protofilaments. About 13 protofilaments make one microtubules. It has been observed. 13 subunits or protofilaments make one microtubule. So in this way, 13 protofilaments make one microtubule, right? Now, it is saying more about the microtubule here. Up to this much you can write. Wall of the microtubule consists of 13 individual linear or spiraling filamentous structure about 5 nanometer in diameter, which in turn is composed of tubulin. Now, the wall of a microtubule, if you see, it has 13 individual linear spiraling filamentous structure about 5 nanometer in diameter, which are made up of tubulin. Each of these filaments, I have said na, is made up of tubulin dimers. Application of negative staining technique has shown that microtubules have a lumen. Lumen means the empty space inside the empty space of 14 nanometer wide and a protofilament or subunit structure in the wall. So let us see the diagram, then you can able to observe, as I have mentioned with you, that about 13 protofilaments, you can see here up to 13, from 1 to 13, up to 13 protofilaments make one microtubule. This is the cross section structure of microtubule from upside down. This is from longitudinal view. If you see the longitudinal view made up of, see, different protofilaments makes one microtubule. Okay, and the diameter of the lumen, the empty space, is about 14 nanometer. Okay. Okay, now let us see the chemical composition of the microtubule. Now each of the protofilament, these are all protofilament, right? These, each of them are protofilament. Like this one is one protofilament, this one is the next protofilament, this one is the third protofilament. In this way 13 protofilaments make one microtubule. About 13 protofilaments make one microtubule, and each of these protofilament is made up of what? Tubulin. Each of these protofilament is made up of tubulin. Now if you see the protofilament here, it has been shown. Okay, each of these protofilament is made up of tubulin dimer, alpha tubulin and beta tubulin, which are assembled at one end and disassembled at the other end, depending on the functions of the microtubule where it is occurring. So uh, biochemically, each protofilament of a microtubule is made up of a protein called tubulin. What is tubulin? Tubulin is an acidic protein. It is an acidic protein. Its molecular weight is about 55,000 Dalton, and it exists in two different forms. Tubulins are found in two types: alpha tubulin, as shown here in green, and the beta tubulin, which shows in brown like color. And each of these tubulin is each of these alpha tubulin and beta tubulin is made up of about 450 amino acids. Okay, now it has been mentioned that these alpha tubulin and beta tubulin must have evolved from the same ancestral protein. Okay, so this tubulin, alpha tubulin and beta tubulin, form in the form of dimers. Okay, they form dimers. So you can say better, they form heterodimers, not homodimers. They form heterodimers. Here you can see one alpha tubulin and one beta tubulin form a dimer. So these heterodimers of tubulins assemble to form a linear protofilament, right? So these heterodimers containing one alpha, sorry, this is not alpha, this one is alpha and this one is beta. So these heterodimers, they form the protofilament one by one, with beta tubulin of one dimer contact with the alpha tubulin of the next. And microtubules are polar in nature. They have a plus end and they have a minus end. Plus end means fast growing end, and a minus end, which are slow growing end, means at one side, on the plus growing end, the tubulin dimers are going to be assembled and whereas on the minus or slow growing end, disassembly takes place. Okay, so microtubules by the help of the tubulin dimer are getting formed from the fast growing end or plus growing end, as here we see in the figure. Here we can see on the plus growing end assembly is occurring. Well, don't see this now. And a minus growing end where disassembly is going to be takes place. Now at the minus growing end or slow growing end, these microtubules are tightly bound by MTOC, called microtubule organizing center. So on the minus growing end, that is here now, it is associated with MTOC or microtubule organizing center. Actually, microtubule organizing center or MTOC protect the minus end of the microtubule from getting disassembly. When this MTOC is not present, then slowly your microtubules getting disassociated. Thus your microtubules getting disassociated by disassembly of the tubulin dimers from the protofilaments. Microtubule organizing center is protecting the minus end of the microtubule from getting disassembly. And these are the sites, these microtubule organizing center are the sites where polymerization of the microtubule occurs. They hold the microtubule so that more assembly at the plus or the positive growing end take place. It protects the minus end, as I have mentioned little bit ago. Where it protects the minus end, that is the slow growing end of the microtubules from getting disassembly. Okay, and the positive or the fast growing ends of the microtubule are protected by the capping proteins. Some capping proteins are there which protects the positive ends of the microtubules. So on the fast growing end or positive growing end, plus growing end, they are protected by the capping proteins, whereas on the negative or the minus or slow growing end of the microtubules, they are protected by the MTOC or microtubule organizing center, which helps in protecting the minus end of microtubules from getting disassembly. Okay. Now let us see about more of the other accessory proteins of the microtubules, which helps in organization of the microtubules. Recently, a number of proteins has been identified that associate with the surface of the microtubules. Not only the tubulin, but other some helper proteins they associate with the surface of the microtubules, and they are called as microtubule associated proteins or MAPS. Now two types of microtubule associated proteins have been observed. Two types of microtubule associated proteins have been observed, which are HMW proteins, and other proteins are also there, tau proteins, TAU. HMW means high molecular weight proteins, which have molecular weight of about 200,000 to 300,000 daltons, whereas tau proteins whose molecular weight is 40,000 to 60,000 daltons. Both these HMW proteins and tau proteins have two domains, means two important functional region. With the help of a domain particular the structure domain, they helps to bind with the microtubules, and the other domain helps to link the microtubules to the other cell components. So again I repeat, these HMW proteins and the tau proteins are the microtubule associated proteins because they bind to the surface of the microtubules. These two proteins, HMW proteins and tau proteins, which bind to the surface of the microtubule, they have two important domains. Domains are the essential regions found within the proteins for particular function. With the help of one domain, this HMW protein and tau protein directly bind to the microtubule. With the help of the other important domain, they link the microtubule with other cell components. Okay. So with the help of other domain, they link with the microtubule with the other cell components. Generally, these microtubule associated proteins tend to speed up the nucleation. It tends to speed up the nucleation step of tubulin polymerization in vitro. It has been observed. In vitro means within the laboratory. Within the lab, we have observed that these microtubule associated proteins helps to speed up the nucleation step of tubulin polymerization. What is nucleation? Nucleation here of microtubule is the process in which several tubulin dimer molecules interact to form microtubule, right? Interact to form microtubule. And this nucleation of microtubule occurs at the microtubule organizing center. Okay, it occurs in the microtubule organizing center. So here about the microtubule associated proteins, what we have discussed here, that microtubule associated proteins, they bind to the surface of the microtubules. Okay, they are very high molecular weight also. As we can see, two different types of microtubule associated proteins are found, HMW proteins and tau proteins. Both of these HMW proteins and tau proteins have two important functional domains, which are the important sites of their protein structure for important function. With the help of one domain, they interact with the microtubule surface, and with the help of another domain, they link the microtubules with the other cell components. With the help of other domain, they link the microtubule with the other cell components. And it has been said that microtubule tends to speed up the nucleation process of tubulin polymerization in vitro. What is nucleation of microtubule? Nucleation of the microtubule is the process by which several tubulin molecules interact to form a microtubule seed. And this step occurs near at the microtubule organizing centers. Okay, I hope you have understood what I have said. Next is microtubule organizing center. As I have mentioned earlier, that microtubule organizing center are the sites where polymerization of microtubules occur. It protects the minus end or slow growing end of the microtubule from getting disassembly. So microtubule organizing centers are the nucleating centers that serve as template for the polymerization of the tubulin. Okay. Microtubule organizing centers are observed in the basal bodies like we have seen in Chlamydomonas, in the centrioles of most animal cells, at the poles of the mitotic spindles, in dividing cells that do not have centrioles, on chromosomes also, like in kinetochores, in the membranes, and probably many other places as well. So microtubule organizing center acts as a nucleating centers. They serve as the site to so that several tubulin molecules interact to form the microtubule seed. Okay, and it protects the end of the minus end of the microtubule from getting disassembly. They are found on various sites, like they are found on basal bodies. In Chlamydomonas we have observed centrioles, most of the animal cells, at the poles of the mitotic spindle, in dividing cells that do not have centrioles, like in most plant cells. On chromosomes also we found this type of microtubule organizing center, like in the kinetochore, in the membranes, and probably many other places as well. So here is the picture to make it simpler to understand about the microtubule associated protein. So you can see one domain of microtubule associated protein interacts with the tubulin of microtubule, while the other domain of the microtubule associated proteins, they can interact with the other proteins, they can interact with the other cell components. Okay. Now this picture that you can see is showing what? It is showing the minus end of the microtubules in each of the following cells, and each of these minus end of the microtubules are embedded within the microtubule organizing center, while the plus ends are often near the plasma membrane. See, the positive end, like this is a interphase cell, as you can see the name given, its positive end are located where? On the near the plasma membrane of the cell, while the minus end are located together, and within this site microtubule organizing center are observed. They are protecting the minus ends of the microtubule. This is a type of ciliated cells having a cilia. The plus ends are protruding towards the near of the plasma membrane, while the minus ends are located inside. Okay, this is a normal dividing cell. As we know, we have drawn a diagrams many times in our junior classes during when we study about mitosis or meiosis. So the positive ends are these ends. These ends are the positive ends where the chromosomes arms are attached. The centrosomes are attached. But the negative ends, the minus or slow growing ends, are actually attached by assembled by the microtubule organizing centers, right? This is a typical nerve cell where you can see the positive end and the negative end. Okay. Now let us see about the assembly and disassembly of the microtubules. How assembly and disassembly is carried out. It has been mentioned here that addition of the tubulin subunits is actually carried out by a GTP hydrolysis and a conformational change. So, with the help of GTP hydrolysis and structure of change in the microtubules, they leads to the formation of addition of the tubulins. Well, I have said about the microtubules before. It is saying here that cytoplasmic microtubules are highly dynamic structures. They are always changing microtubules, constantly forming and disappearing depending on the cell activities. What type of activity cell is performing, depending on that the microtubules are constantly changing in their structure. They are like microfilaments, grow by reversible addition of subunits accompanied by nucleotide GTP hydrolysis and conformational change. On one side, on the positive growing end, they are constantly adding the tubulin, accompanied, that means help with the, and it is carried out by GTP hydrolysis and conformational change as well. The process of polymerization, that is assembly, and depolymerization, that is disassembly of microtubules, appear to be form of self assembly. So they self assemble actually. Now where the assembly of microtubule occurs, it is occurring through a program process actually. Within the cell, the sites of orientation are the MTOC from which the polymerization is directed. So obviously polymerization of the microtubule occur at the microtubule organizing center. Okay, as I have said that these are the MTOC are the sites where polymerization of the microtubule occur. right? And it will be helped by GTP hydrolysis and obviously conformational change in the microtubule. Now, the quantity of polymerized tubulin is high at interface. Now, this addition of tubulin from the minus end to the positive end, its, its rate is very high during the interphase but low at prophase and anaphase. Okay. Now, within the cell, microtubules are equilibrium in the with free tubulin. Normally, within the cell, these microtubules are in equilibrium with free tubulin. Phosphorylation of the tubulin monomers is catalyzed by a cyclic AMP dependent kinase favors the polymerization. Now, normally, what happened? I have discussed. I have said the structure of alpha tubulin and beta tubulin, which forms a tubulin heterodimer. Phosphorylation of the tubulin monomer is occur. Initially, they do not bind in that way. Normally, they have to be phosphorylized. They have to be phosphorylized by the help of a kinase called cyclic AMP dependent kinase, which helps in polymerization. Okay. A definite relationship has been found between cell shape, number, and direction of microtubules and cyclic AMP. It has been observed that phosphorylation of the tubulin monomers is done by some kinase, cyclic AMP dependent protein kinase, which helps in polymerization of the tubulin monomers to form tubulin dimers and then it associate to form protofilaments. Now, in a microtubule, the assembly of tubulin dimers take place at one end, while disassembly is common at the other end. Right? Now, within the microtubule, assembly of tubulin dimers take place at one end, fast growing end, positive end that we know, and disassembly is common at the other end, which is the minus growing end or slow growing end, where the microtubule organizing centers are holding firmly so that disassembly does not occur. Now, suppose if we treat that cells with certain drugs like colchicine, vincristine, or vinblastine, then assembly of the microtubules will be inhibited while disassembly will continue, leading to disorganization of the microtubule. Now, if we treat the cells with colchicine, vincristine, and vinblastine, then what will happen? The positive growing end of the microtubules will be blocked while disassembly will continue from the minus or slow growing end. That means MTOC becomes non-functional at that time, leading to disorganization of the microtubules. Okay. Further disassembly is accompanied by hydrolysis of a GTP molecule. Okay, and lack of GTP stops the assembly. Further, assembly is accompanied or helped by hydrolysis of a GTP molecule. For disassembly, I have said we can use drugs like colchicine, vincristine, and vinblastine so that the positive growing ends of the microtubule is inhibited. Okay, while disassembly continue. But for the opposite case, if you want to allow assembly of the microtubules, it can be accompanied by hydrolysis of a GTP molecule, which will hydrolyze to form GDP. And it has been mentioned that if GTP activity, if hydrolysis of a GTP does not occur, then it also interfere with the assembly of the microtubule formation. In vivo, control of assembly and disassembly of tubulin involves calcium and calcium binding protein called calmodulin. It has also been observed that for the assembly and disassembly of the tubulin dimers. It also involves the calcium ion and calcium binding protein called calmodulin. Addition of calcium inhibits polymerization of tubulin. This effect is also enhanced by addition of calmodulin. So many factors plays an important role for the assembly and disassembly of the microtubule. As I have said that GTP activity, hydrolysis of a GTP is required for the assembly of a microtubule. Second, certain drugs, if we treat the drugs with the tumor of microtubule, then the microtubule assembly will get disrupted, like colchicine, vincristine, vinblastine. These drugs, they assembly, they prevent the assembly of the microtubule. They actually interfere with the fast growing end or the positive growing end of the microtubule. Thus the microtubule disassembly continues. Third, I have said about the kinase enzyme which cause phosphorylation of the tubulin monomers to form tubulin dimers. So a cyclic AMP dependent kinase helps in phosphorylation of the tubulin. Fourth, calcium and calcium binding protein calmodulin also helps in your assembly of the tubulin monomers. Okay. Okay. So more it has been mentioned here. Let us see. I am going here. Now, in vivo, it has been observed in vitro, sorry, not in vivo, in vitro, that means within the laboratory, it has been observed that how polymerization of tubulin occurs. Okay. In vitro polymerization evidently involve two distinct phases. So two distinct phase has been observed when we polymerize the tubulin dimers to form microtubules. One is the initiation phase and other is the elongation phase. During the initiation event, it seems to involve formation of some multimeric nucleating center, just like microtubule organizing center are getting formed during the initiation phase. Okay, during in vitro polymerization of microtubules, the alpha and the beta tubulins combine to form heterodimers. Initially, what happened? Alpha tubulin and the beta tubulin combine to form a heterodimer, and that is possible by phosphorylation of the microtubule tubulin monomers by the help of cyclic AMP dependent protein kinase, which helps in polymerization. Side by side assembly, wait for a minute. Okay, these heterodimers now assemble to form ring or spiral like structure. So this heterodimer associated to form multimeric ring, spiral in structure, and other intermediate structures which eventually opens up to form protofilaments. Now what happens? One by one these heterodimers join because they have been phosphorylated. They join side by side to form ring-like structure, spiral-like structure, which are observed in vitro within the laboratory, and finally they opens up to form a strand of protofilament. Side by side assembly of this protofilament creates sheet-like structure that curls. That curls means they curl up to form a protofilament, to form a tube-like structure. So these heterodimers side by side they form protofilaments, and these protofilaments then curl off side by side and create a sheet like structure. First they form a sheet like structure and then this sheet like structure curled up, okay, to form a tube like structure. Elongation of this short cylinder occurs by direct addition of new heterodimers at one end of the tube, that is plus end. The elongation occurs, and it is believed that during anaphase, addition of dimers to one end of the microtubule is accompanied by loss of dimer of the other end. So here you can see the whole thing which has been mentioned, the alpha and the beta tubulin here which are assembled by phosphorylation. They has been phosphorylated by a help of a kinase enzyme. Okay, cyclic AMP dependent kinase, and these heterodimers then assemble to form various patterns of structure. Okay, that's we can see, and finally they form a linear or protofilament. This protofilament form a sheet like structure first, okay, like a sheet of paper. Now this sheet of paper, if you imagine, they curl up, they bend to form a hollow tube like structure to form a microtubule. Okay, now there are various functions of the microtubule. As you know, various functions are there within the microtubule. What are the functions of the microtubule? uh, if I say about the function, first is the mechanical function. The shape of the cell, like red blood cells of non-mammalian vertebrates and some cell processes of protuberance. Protuberance means a bump like projection, a part of the cell like action, like dendrites that we found in the nerve cell, just like projection like structure. In macrophage, we also see, right? So this type of projections are actually formed by the microtubules. Okay, and it is correlated with the orientation and distribution of the microtubules. So the first is the mechanical function, which determines the shape of the cell. So microtubules determine the shape of the cells, like red blood cells of non-mammalian vertebrates. Okay, then some cell process having protuberance like structure, bump like projection, part of the cell, like in actions, like in dendrites of nerve cells, microvilli, which has been correlated with the orientation and distribution of the microtubules. Second is morphogenesis. Microtubules actually used to determine the shape of developing cells. Microtubules is used to determine the shape of the developing cells, like enormous elongation in the nucleus of a spermatid that we observe during spermatogenesis, which help by production of a arrangement of microtubules wrapped around the nucleus in a double helical arrangement. So we have seen a enormous elongation in the nucleus of a spermatid during spermatogenesis, helped by the production of an arrangement of microtubules which are wrapped around the nucleus in a double helical arrangement. So microtubule is actually helps to determine the shape of a developing cell during cell differentiation. Like I have said, during spermatogenesis, we see the enormous, huge nucleus elongation is observed in the sperm. Uh, and it occurs due to the orderly arrangement of the microtubule that wrap around the nucleus in a double helical arrangement. Then obviously, polarity and motility helps in the movement also. Microtubule helps in cellular movement through gliding. So by the gliding movement, that is movement without fembrae, movement without cilia, movement without flagella or pili, they can able to move. So to determine the intrinsic polarity of certain cells is also related to the microtubules. Directional gliding of the culture cell is found to depend on microtubules. It has been observed within the laboratory that cells glide without having any fembrae, without having any flagella, and this gliding move type of movement occurs is due to the help of microtubules. Contraction, microtubules helps in spindle fiber contraction. Spindle fiber is also made up of microtubules. Microtubules plays a major role in the contraction of spindle fiber and movement of chromosomes and movement of centrioles, as well as in ciliary and flagellar movement. So microtubules also helps in contraction of the spindle fibers. It also helps in movement of the chromosomes. It also helps in the movement of the centrioles, as well as in ciliary and flagellar motion. The cilia and the flagella, which helps the cells to move, bacterial cells, uh, Okay, they are also made up of microtubules. So micro also helps in movement. And last is circulation and transport. Microtubules also helps in transport of macromolecules, granules and vesicles within the cell. Microtubules helps in movement of the macromolecules, movement of the granules and vesicles within the cells. Many examples are there, like if I say the first example, the protozoan Actinosphaerium heliozoa sends out a long thin pseudopodia within which cytoplasmic particles migrate back and forth. So pseudopodia, as you know, they are temporary arm-like projection of a eukaryotic cell membrane that are developed in direction of movement, and it mainly consists of actin filaments and microtubules. So with the help of pseudopodia, they the cytoplasmic particles migrate, they move back and forth. Okay. Then the next example, in the protozoa nasula, microtubules drive the food in the gullet. Okay, so the protozoa nasula, they have microtubules which helps in movement of the food, passage of the food from mouth to the digestive tract is done by the microtubules found within the nasula. So the microtubules helps in passage of the food here in the protozoa called nasula. Okay, so these are the some examples related with the microtubules. Then if I say the number fourth, uh, the erythropores which are found in the fish scales or in pigment granules may move at a speed of 25 to 30 micrometer per second within the microtubules. Erythropores are the pigment, uh, they are the pigment cells that can shift the body, uh, they can shift the body, they move, they have their pattern movement actually, and they are found within the fish scale pigment granules. They can move at a speed of 25 to 30 micrometer per second between the microtubules. So many functions are there for movement of the macromolecules, for movement of the cytoplasmic particles can be done by the microtubules. So this was all about the microtubules. I hope you have understood what I have mentioned with you now in these last 50 minutes. Um, okay.